Hello friends, today we are going to discuss the two phases of glycolysis. When glucose break down into the two molecules of 3 carbon pyruvate, at that time the different types of enzymes are involved in this different phases. These enzymes they can convert the 6 carbon glucose into two molecules of 3 carbon pyruvate. So this glycolysis means it is an lysis means breakdown of the glucose. But in this breakdown, the different phases are there. Into that, two important phases are phase first, that is a preparatory phase, and phase second, that is a payoff phase. When the different enzymes are act on the glucose, these glucose molecules they get converts into the two molecules of 3 carbon pyruvate. The specific enzyme catalyzed reactions being bifurcated or categorized into the two phases. These are preparatory phase and payoff phase. Preparatory means a specific glucose they will be prepared for the payoff phase, or they will be when in each and every metabolic pathway, or in case of the most of in catabolic pathway, the specific microorganism they can gain the energy from the breakdown of the specific biomolecule into the smaller particles, and then they will be generate the ATP molecules, and these specific ATP molecules these are used as a energy currency. So for this preparation of this ATP or adenine triphosphates, because each and every cell they have to require the energy in the form of ATP, GTP, CTP, or different types of the triphosphates are there. So in case of the glycolysis, the same process are carried out for the preparation or for the generation of ATPs. But the specific glycolysis they can consist the two phases, preparatory phase and payoff phase. Because in preparatory phase, the specific cell or specific microorganism they will be invest the specific ATPs or specific number of ATPs, and then in case of the payoff phase, they will be refund it from the same protocol or from the same process. So in this case, let's we discuss the preparatory phase. The preparatory phase it consists the first 5 steps of glycolysis. Basically, total 10 phases are there in case of 10 steps are present in case of the glycolysis. Into that first 5 steps are considered as a preparatory phase. In this specific first 5 steps we have glycolysis in which the glucose is enzymatically phosphorylated by ATP to yield the fructose 1,6 bisphosphate. In this first phase or first step, the specific glucose get firstly converted into the glucose 6-phosphate. Then the specific glucose 6-phosphate get converts into the glucose 1,6 bisphosphate, and this glucose 1,6 bisphosphate get converts into the fructose 1,6 bisphosphate or diphosphate. So this fructose 1,6 bisphosphate is then split into in half to yield two molecules of 3 carbon compounds or 3 carbon containing molecule, that is a glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, because with the help of enzyme aldolase. The fructose 1,6 bisphosphate is then split into the two molecules, because basic glucose it is a 6 carbon and pyruvate or the glyceraldehyde 3 phosphate and dihydroxyacetone phosphate, these are the 3 carbon compounds. These the specific in this step, the glyceraldehyde 3 phosphate it is a aldehydic sugar, and dihydroxyacetone phosphate it is a ketonic sugar, because basically the basic or the parent sugar that is a fructose it is a specific fructose it is a ketonic sugar. So it consists the C double bond O, that C double bond O containing three molecule carbon compound, that is a dihydroxyacetone phosphate, and the aldehydic group containing aldehydic group containing 3 carbon compound, that is a glyceraldehyde 3 phosphate. After the action of aldolase, they will be converted into these two compounds, that is a glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Then thus the first phase results into the cleavage of hexo chain. In this phase, first phase or that is a preparatory phase, the cleavage of hexo chain will be takes place. The six-membered glucose get converted into the 3 carbon dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. This cleavage requires an investment of 2 ATP molecules. This cleavage requires an investment of 2 ATPs to activate the glucose molecule and prepare it for its cleavage into the 3 carbon compound. So first glucose is invested when the glucose get converts into the glucose 6-phosphate, and second ATP invested when the glucose 6-phosphate get converts into the glucose 1,6 bisphosphate or glucose 1,6 diphosphate. To that, this is an glucose. Glucose it is an hexose sugar. With the help of hexokinase, the one ATP is invested into this step first, and this glucose get converted into the glucose 6-phosphate. Then the phosphohexose isomerase, the isomerization of this specific sugar, this glucose 6-phosphate get converts into the fructose 6-phosphate with the help of enzyme phosphohexose isomerase. Then this fructose 6-phosphate get converts into the fructose 1,6 diphosphate, and also they have to invested the second ATP. First ATP is invested at the step first, and second ATP is invested at the step third. Then this fructose 1,6 diphosphate or fructose 1,6 bisphosphate with the help of enzyme aldolase, they will be converted into the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Dihydroxyacetone phosphate it is a ketonic one, and diglyceraldehyde 3-phosphate it is a aldehydic one. So in this phase, or it is in preparatory phase, there are the 5 steps are involved in it. The glucose get converts into the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Then, in case of the payoff phase, this phase constitutes the last 5 reaction, or the after 6, 7, 8, 9 and 10 last 5 steps of glycolysis, these are considered as a payoff phase. This phase marks the release of ATP molecules during the conversion of glyceraldehyde 3-phosphate to 2 molecules of pyruvate. Because glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, these are 3 molecule compounds, or 3 carbon molecules are formed because of the action of aldolase in initial reaction into this step. The dihydroxyacetone phosphate and glyceraldehyde 3-phosphate with the help of enzyme aldolase. So then the specific for here 4 molecules of ADP are phosphorylated to ATP. The 4 molecules of ADP are phosphorylated to ATP. Although these 4 molecules of ATP are formed, then the net result is only 2 molecules of ATP per molecule of glucose oxidizer, because since 2 molecules are utilized in the first, because initially the specific cell or bacterial cell, they will be invested the 2 ATPs, and then in the payoff phase, this two the specific cell they will be gain the 4 ATPs. So net gain is that is a 2 ATPs only. The first 2 ATPs are invest basically in case of the payoff phase, the cell can get the 4 ATPs, but initially the specific cell they have to invested the 2 ATPs. Then in this step, the specific it is a step number, or step number 6 of glycolysis. The triose phosphate dehydrogenase. It is an enzyme. They will be convert the glyceraldehyde 3-phosphate into the 1,3 disphosphoglycerate. It is an anhydrate or acetate of this specific glyceraldehyde 3-phosphate. In this, they can got the one NADH. NAD get converts into the NADH2. 1 3 disphosphoglycerate. This 1 3 disphosphoglycerate they will be converted into the 3 phosphoglycerate. This 3 phosphoglycerate they will be formed because of the enzyme phosphoglyceric acid kinase. Phosphoglyceric acid kinase plus cofactor Mg double plus. Then this 3 phosphoglycerate get converts into the 2 phosphoglycerate with the help of enzyme phosphoglyceromutase. The mutase enzyme they will be converts the 3 phosphoglycerate into the 2 phosphoglycerate. These 2 phosphoglycerate get converts the specific converted into the phosphophenol pyruvate, and the enzyme that is the enolase. Enolase it is an enzyme act on the 2 phosphoglycerate, and these 2 phosphoglycerate get converts into the phosphophenol pyruvate. This phosphophenol pyruvate they get converts into the pyruvate. At that time they will be converts the ADP into the ATP, and they will be get the ATP. In this conversion of 1,3 disphosphoglycerate to 3 phosphoglycerate, there will be gain of 1 ATP, and also into the last reaction phosphophenol pyruvate get converts into the pyruvate. At that time also the cell can get the 1 ATPs. So total net gain from the 1 diglyceraldehyde 3 phosphate, that is a 2 ATPs. Also, because of the 6 carbon molecule of glucose, they can split into the 3 carbon compounds, that is a dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. Also, this dihydroxyacetone phosphate they get converts into the glyceraldehyde 3-phosphate with the help of the phosphotriose isomerase. Because of this isomerization, the glycerol dihydroxyacetone 3-phosphate into the glyceraldehyde 3-phosphate, and this glyceraldehyde 3-phosphate also, the glyceraldehyde 3-phosphate also, they will be go through this same reaction, and they will be converted into 1,3-disphosphoglycerate to 3-phosphoglycerate, 3-phosphoglycerate to 2-phosphoglycerate, and 3-phosphoglycerate to phosphopenol pyruvate, and lastly the phosphopenol pyruvate get converted into the pyruvate. So the two ATPs from the dihydroxyacetone phosphate and two ATPs from the glyceraldehyde 3 phosphate. So total net gain in case of the payoff phase, that is a 4 ATPs, but the same cell they have to invested the 2 ATP in case of the preparatory phase. So there is a only the net gain of ATP, that is a 2 ATPs from the one glucose molecule. Okay, is that clear? If you have any doubts regarding the glycolysis, they will be post into the comments, or otherwise they will be we will you will be post the specific questions into the WhatsApp group.